All right. So, welcome back to part two of our cells lecture. So, in this part, we're going to continue looking at some of our biomolecules, including lipids and carbohydrates, and then we'll go into a little bit more detail on our proteins as enzymes and catalysts and things like that. Okay. So, our third biomolecule are lipids. Lipids are what we kind of generally refer to as fats, and unlike proteins, nucleic acids, and carbohydrates, there's not a single monomer for every type of lipid. Okay, so they're going to vary a little bit. All right. So one type of lipids are called triglycerides. Okay, so triglycerides contain something called a glycerol. They also contain three what are called fatty acids. Okay, so triglycerides contain three fatty acids, and this is what we see here on the left. All right, so on this kind of upper left part of what we're looking at here. So there are three fatty acids. This is what we typically use for long-term energy needs and long-term energy storage. So whenever you're pulling on your fat reserves and burning those fat cells, what you're burning are triglycerides. Okay, so they can provide lots of energy. So they provide 9 kilocalories per gram, whereas proteins may only provide 3 or so. All right. Next one are called phospholipids. Okay, phospholipids are, and we'll see this later in this lecture. Phospholipids are extremely important for what we're going to be talking about. All right, phospholipids are the main component of our plasma membrane. Okay, they're not the only component, but they're the main component. All right. Now, phospholipids have two general parts. Okay, they have a head part and they have a tail part. All right, this head is what's called hydrophilic. All right, hydrophilic meaning it is attracted to water. It likes to be in contact with water. Hydrophobic meaning the opposite, right? It has a fear of water. It is repelled by water. All right. The head of these phospholipids is primarily a phosphate group. Okay. But they could also have this glycerol, like we talked about over here with the triglyceride. And then the tails are fatty acids. Okay. And now you note on the fatty acids on this phospholipid molecule here. One is saturated and it is straight. One is unsaturated and it is kind of bent and crooked. All right. We'll talk about this more later and how this influences how fluid the membrane is. All right. The next one is a steroid. All right. So steroids can be used for chemical messengers. You'll see when you get to the endocrine system in AMP2 that there are hormones, so the chemical messengers of the endocrine system. Some of them are steroid based. Okay. Steroid is a type of lipid. Our plasma membrane is made of phospholipids. And so the interaction between steroid hormones and the plasma membrane is one that allows the steroid hormone to pass freely through the membrane. Okay, because there's a common principle in chemistry that like dissolves like. Similar things can be dissolved that are similar to it, and so a lipid membrane can dissolve a steroid or lipid-based hormone. Okay. And then you can also have things like wax. And a good example of this in anatomy is cerumen, okay, or earwax, right? So the ceruminous glands in your ear can produce earwax. This can line the inside of your external acoustic meatus, or what we generally think of as our ear canal, and it can provide some protection in that way. Okay. So whenever we continue here, if we look a little closer at our phospholipid, we said earlier that it has a head, it has a tail. The head is hydrophilic, meaning it likes to be in contact with water. The tail is hydrophobic, meaning it does not like to be in contact. Now, for our plasma membrane, we'll have two layers of these phospholipids, all right? And when you have two layers of this, we call this a phospholipid bilayer, okay? So our plasma membrane is a phospholipid bilayer. It is two layers of phospholipids opposed against each other. The heads are always going to be oriented away from the inside of the cell, and the um, um, or one layer is going to be oriented to the outside of the cell, one is going to be oriented to the inside, and then the tails are going to be oriented away from both the inside and the outside of the water. So they're going to be opposed to each other in the middle, right? So this interaction of the two phospholipids has to do with the fact that this molecule is called amphipathic. Okay, amphipathic meaning it has a polar region and a non-polar region. So there's a region that is attracted to water, and there's a region that is opposed or repelled by water. All right. The polar region dissolves in water. This is the head. All right. So they have a polar head. The non-polar region is repelled by water. This is the tail. All right. So we have hydrophobic non-polar tails. We have hydrophilic polar heads. And again, they form this kind of bilayer. All right. But again, keep in mind, the phospholipid is not the only thing in the plasma membrane, but it is the main thing in the plasma membrane. Okay. We'll see later that we'll have different proteins. So one of our biomolecules, protein channels embedded within this plasma membrane here. All right. And then lastly, our last biomolecule that we'll talk about are carbohydrates, right? So carbohydrates are made up of their monomer, which are sugars, so just a single sugar monomer. And this is going to be a monosaccharide, okay? So if you think of something like glucose, if you think of something like fructose or galactose, these are all examples of a monosaccharide. Okay, the term mono means one, saccharide refers to sugar, okay? If you put two of them together, you get a disaccharide, so something like sucrose, something like lactose, right? So milk sugar, these are all disaccharides. And then when you put lots and lots together, essentially more than three, but usually it's tens and hundreds, you get a polysaccharide, right? So poly means many, saccharide sugar. So this is many sugars or many monosaccharides, right? The example of this in humans and in us is something called glycogen, okay? Glycogen is essentially strands in these branching networks of tens and hundreds of glucose molecules that can be stored in the liver, that can be stored in muscle cells. And then whenever we need them, or whenever we need to pull on our glycogen reserves and increase blood sugar, we can break down glycogen, right? And we'll talk about that maybe a little bit in AMP1. You'll talk about it a little bit more in AMP2, right? So whenever we want to break down this glycogen, we can go through a process called glycogenolysis. Okay, so if you think of the term glycogenolysis, there's two words. There's glycogen, and then there's lysis. Okay, glycogen refers to the glycogen, right? So the polymer. Lysis means to break or to split up or to break down. Okay, so glycogenolysis is the splitting up or breaking down of glycogen into glucose, right? You can then go the opposite direction and go from glucose to glycogen. In this case, this is called glycogenesis. Okay, so glycogen, glyco refers to glycogen, and then genesis means the beginning of, or the formation of, or the creation of, right? So this is the formation or the creation of glycogen from glucose. Okay, and as we said earlier, we can store this in our liver and our muscle cells, and then under the influence of various hormones, maybe like growth hormone or thyroid hormone or cortisol or something, we can pull on our glycogen reserves and increase our blood sugar to then be used for cellular respiration to make ATP so we have energy. All right. Okay. Now we can also add things and modify carbohydrates as well. So you can sometimes add a lipid molecule or a lipid component to a carbohydrate, right? And when you add something like this to a carbohydrate group, this is called glycosylation. Okay, and glycosylation can allow different cells to interact with each other. It can also serve as something like an identification tag. So essentially, this cell is alerting other immune cells and other cells around it that this is a native self cell and it is not foreign and it does not need to be destroyed or anything like that. Okay, we'll talk more about glycosylation in just a little. And then we know that we can use carbs for more short-term energy in the body, whereas lipids are going to be used for a little bit more long-term energy. So we can break down carbs into sugars and then use those sugars to make ATP. Right? So answer this question. Damage or dysfunction to which of the following monomers would disrupt the synthesis of carbohydrates, right? So pause the video, try to answer the question. I'm going to move on. So the correct answer in this case is going to be C, right? So the key here is we're looking at carbohydrates, right? So fatty acids, this is a monomer. So this question is essentially asking for the monomer of carbohydrates. Fatty acids, that's a monomer, but not monomer for, or you can think of it as a monomer, but not a monomer for carbohydrates. This is associated with lipids. Amino acids are the monomers for proteins. Monosaccharides is the correct answer. It's the monomer for carbohydrates, so single sugar, right? And then glycogen, this has to do with carbohydrates, but glycogen is not a monomer, right? Glycogen is a polymer, so that would be incorrect. So going back and looking at our proteins, proteins can function as enzymes and catalysts, right? So this is going to kind of lead us up into our discussion of protein synthesis, all right? But we're going to talk about proteins as enzymes and catalysts. So a catalyst or an enzyme functioning as a catalyst is a protein that speeds up the rate of some type of chemical reaction, okay, or different cellular reactions, right? What it does is it decreases something called the energy of activation or the activation energy, right? So think of the activation energy. Imagine you are trying to push a large boulder down a hill, okay? But you're at the bottom of the hill, but you want to push it down the hill, all right? So in order to push it down the hill, and you're at the bottom of the hill, you've got to get to the top of the hill, right? So you have to put in some effort, put in some energy to get yourself and the boulder to the top of the hill, right? That in this instance would be the activation energy, okay? You've got to put energy into the equation, into the reaction, in order to get your net result, which in this case would be pushing the boulder down the hill, right? So enzymes functioning as catalysts, and again, remember, enzymes are proteins, okay? They can decrease the energy of activation, okay, or the activation energy. So in this case, they may lower if we think of our kind of hill with the boulder example, they may decrease the size of the hill, right? So you still have to put in some energy, but you have to put in less, right? You put in less, you can get your result, you can get the pushing of the boulder down the hill, you can get your result more quickly, right? And so that's the idea here of our enzymes. They're speeding up chemical reactions, okay? That's very important. All of our enzymes, when you're in AMP1, when you're in AMP2, you're reading in the book, you're watching a video, you're doing whatever, enzymes end in ASE, okay, ACE, okay? So protein kinase, thyroid peroxidase, proteases, lipases, okay? These are all examples of different proteins. And these are examples of enzymes. Different enzymes have different functions, all right? A kinase is something that phosphorylates. It adds a phosphate group to something. Thyroid peroxidase, you talked about this in AMP2. This is something that helps make our thyroid hormone. Proteases break down proteins. Lipases break down lipids, okay? Our enzymes are going to have something called an active site, and they're going to bind something called a substrate, right? And so we'll look at that here in just a little bit, right? So this is a nice schematic and a nice graph showing the functionality of an enzyme as a catalyst in decreasing the activation energy, okay? So what you have is you have your starting point, okay? This dotted line would be without an enzyme or no catalyst. The solid line would be with an enzyme as a catalyst, right? Now, what you see, or what you hopefully see, is that the net result of this reaction is the same whether you have a catalyst or you don't have a catalyst, okay? The net result is the same. If we drew a straight line from where we started, straight over, okay, we still get the same result, same net result. But with an enzyme acting as a catalyst, we get that result more quickly, right? Because we don't have to invest and put in all this energy. We only have to put in just a little bit, right? So different chemical processes and different reactions will utilize enzymes so they can proceed more quickly. So a good example of this is the process of cellular respiration, right? There are lots and lots of enzymes that end in ASE that help with the breakdown of glucose and the creation of ATP, right? If you did not have these enzymes, it would be extremely difficult and it would take an extremely long time to make our ATP and to make our energy, okay? So when we look at these enzymes, the way they function is they have, as we said previously, they have something called an active site. Okay. And when you think of this active site and this substrate, think of almost like a lock and a key. Okay. The lock is the enzyme. The cutout in the lock for where the key fits, that's the active site. And then the key coming in, that is the substrate. Okay. So its active site is going to be very specific, very unique, and it's going to fit and bind only that one substrate. Okay. The substrate can come in, bind to its active site. Sometimes it can undergo a slight change in something called an induced fit, and we'll talk about that in the next slide or two. And then you get some reaction proceed. Okay, so they may join two smaller molecules together. They may split things apart. In the process of cellular respiration, we see that we're breaking glucose down. So we're splitting things apart. All right. The important thing here is that an enzyme is able to continue functioning after it acts on a particular substrate. So it's not a one and done type of molecule. It can act on a substrate, unbind that substrate, and it's ready to bind the next one. Okay. And then, as we said earlier, you can alter the functionality of these enzymes by altering acidity level, temperature, other things like that, right? So this is why maintaining a proper blood pH in your body. So the normal blood pH is 7.35 to 7.45. Maintaining a proper blood pH is extremely important for chemical processes in the blood, okay, and in the cells. This is why having an extremely high temperature or having a really high fever is also very dangerous. You can denature the proteins, okay? So we said a second ago that sometimes they can undergo what's called an induced fit. All right. So this is when the substrate, the active site of the enzyme slightly shifts and slightly changes shape to better accommodate the incoming substrate. All right. So if you look here at this example to the far left, notice this kind of little raised ridge here. It kind of looks rounded. All right. But if you look at the corresponding region where it's going to fit, this part is more or less pointed. Right. So when this substrate comes and binds, this enzyme with its active site can slightly change shape or undergo this induced fit to better accommodate the incoming substrate so they can undergo whatever process it needs to be. And in this case, it's breaking this thing down. Okay, so it's releasing two new products into the cell or into the blood or wherever it may be. Okay, so you have a substrate come in and it binds very nicely with an enzyme. Okay, so this is an oversimplified schematic and example here, but it is a nice illustration as to what the enzyme is, where the active site is, and then how the substrate comes in and fits. All right, and then here are some videos on induced fit. So it turns out that as you alter the environment of an enzyme, you alter its functionality, right? So as we said, alter the pH, alter the temperature, chemicals or toxins in the environment, right? So in general, as you increase the temperature of the environment, you increase the activity of the enzyme, okay? But you may be thinking, but we just said previously that if we increase the temperature too much, we can denature the protein. That is also true, right? So what that means is there is a kind of a medium, there's a happy medium that has to exist between increasing the temperature too much and not having it be at the right level, right? So this is showing this top graph. As we increase the temperature, we are increasing the activity or the rate of the enzymatic function, right? So increased temperature, this is showing we kind of exponentially begin to increase the rate of activity, right? This middle graph is showing that we may have a constant temperature in which the protein is not being denatured, and then at a certain temperature we have this large drop off because the protein is denatured, right? And then this bottom graph is putting these two together. So there's a nice happy medium when you overlap these two graphs that has an optimal temperature for an optimal rate of enzymatic activity. Okay, so all this is saying, and all you need to realize from this, is that you can't just continually increase the temperature of the environment and have that enzyme continuing to increase in activity. Okay, you get to a certain threshold where activity drops off because it's denatured and you lose the functionality. Okay. All right. So if we look here at this example, this is a wonderful example of an enzyme acting on a substrate, right? So some of you may be lactose intolerant, right? If you're lactose intolerant, you know that you can't have generally dairy products, right? You can't have things with lactose sugar, okay? So lactose is a sugar, right? It ends in OSE. And the reason you are lactose intolerant, okay? Sometimes this condition is also called lactase non-persistent, okay? Um, is that you do not have a particular enzyme called lactase, okay? So in this case, this is lactase. I spelled lactase wrong. There's not two A's, there's just one A. So ignore that. There's only one A in that lactase. But it ends in ASE, okay? So what happens here is because you don't have lactase, you can't break down lactose. Lactose is a disaccharide. We cannot absorb disaccharides in the body. We can only absorb monosaccharides. So you drink milk or you have cheese or you have, something that has lactose in it, and you're lactose intolerant, you get the GI discomfort, you get the gastrointestinal pain, you get all of the things that are associated with being lactose intolerant and consuming lactose because you don't have that particular enzyme. Okay. So lactose is a disaccharide. It's composed of a glucose and a galactose. Both of those are monosaccharides, right? So two monosaccharides together, you get a disaccharide. If you have the enzyme, right? If you are lactase persistent, you have the enzyme. Then when you bind lactose, you're able to break it down into its two constituent parts, glucose and galactose, and then we can absorb in the GI tract and we can actually use those, okay? So it's actually not normal if we think of it from a biological perspective for mammals. It's not normal to be able to digest lactose. Okay, most mammals lose the ability to break down and digest lactose after childhood. Okay, so people who are you can kind of think of it like this: people who are lactose intolerant are actually kind of the normal ones, right? Everyone else that is able to digest lactose and break it down actually has a mutation and has a variant that allows them to be able to do that, right? So this is an example of a mutation, a genetic mutation actually being beneficial, okay? So there's in kind of common speak and common discussion, you think of a mutation like, oh, they have a mutation or that's a mutant or something like that, right? It has a very negative connotation. But all mutations are not necessarily negative. Okay, it's dependent on the individual that the mutation happens to. It depends on the environment that that individual lives, and it depends on the effect of that mutation. Right? Does it increase their likelihood for survival and reproduction, or does it not? Okay. All right. So here's another video on the induced fit model that you can look at. Okay. All right, so we're going to stop here and we'll pick up with part 3, talking about biological membranes, and then we'll eventually get into protein synthesis.